Students, in this class we are going to see some of the important diseases which are affecting the cotton and causing economic yield loss, and which includes the Fusarium wilt, which is caused by Fusarium oxysporum palmaris vas infectum, and then verticillium wilt caused by Verticillium dahliae, and then root rot, anthracnose, grey mildew, and the leaf blights caused by Alternaria as well as Myrothecium, Cercospora. Besides the bacteria which is also causing leaf blight, and the recent entry, that is the tobacco streak virus, which is causing diseases on cotton plant, including the parawilt, which is earlier said to be the physiological disorder, and now with the important fungal pathogen have also been reported for this parawilt disease. And let us see the diseases one by one and how to manage the disease under the field condition. The Fusarium wilt, which is caused by Fusarium oxysporum, form of Specialis vas infectum. The seedlings, it affects from the seedlings, and the cotyledon, which turn into yellow and then completely brown, and the base of the petiole there will be a brown ring formation, which ultimately results in wilting and drying of the affected plants. In the grown up plants, the yellowing start from the edge, and the area around the vein, which is showing the discoloration from the margin and spreads towards the midrib, and ultimately the leaves lose their turgidity and gradually turn into brown and then finally droop down. And on the older leaves, it starts at the base, followed by it starts from the older leaves, and it moves towards the upper part of the plant. Defoliation and complete wilting of the affected plant are the important symptom notified under the field condition. And when you take the longitudinal section of affected plant, you could see the browning or blackening of vascular tissues which are extending upwards on the branches, and the affected plant in the late season become completely stunted with only fewer bolts, other two very small and open before they mature. And this is the typical symptom of the pathogen. There is yellowing of leaves, and then this is the field, affected field, showing patches of emptiness due to wilting of the plant. And when you take the cross longitudinal section, it shows the internal discoloration, vascular browning or blackening due to infection by the Fusarium. And this Fusarium is known to produce three types of spores: macroconidia, microconidia, and chlamydospores. This macroconidia, which is of sickle shaped and 1 to 5 septate, hyaline and thin walled, which tapered at both the end, which is falcate in shape, and then microconidia, which are hyaline, thin walled, spherical or elliptical, mostly single celled and rarely of two celled. It also produces the chlamydospores, which are dark colored, thick walled, and it may be either terminal or intercambially. Besides that, the fungus is also known to produce a vivotoxin, fusaric acid, which is also partially responsible for the wilting of affected plants. And this is the affected plant, and this is the typical shape and septation of macroconidia, and this is microconidia and chlamydospores. And the soil temperature of the generally the Fusarium is will be severe where the high soil temperature and low soil moisture prevails. So in the hot and dry periods followed by rainfall, where the soil temperature is of 20 to 30 degree, the incidence will be heavy, and the incidence will be more in the black cotton soil with an alkaline reaction. And increased dose of nitrogen as well as phosphatic fertilizers favors the development of disease. Besides, the wounds caused by the Melidogene incognita nematode as well as the grubs of Asheville Myeloceras postulatus also favors the pathogen to enter and establish in the plant. And the fungus can survive in the soil as well as in the infected plant debris as chlamydospores, and the pathogen is both externally as well as internally seed-borne. The primary infection is mainly from the dormant mycelium as well as the chlamydospores present in the infected plant debris as well as in the soil, while the secondary spread is mainly through the conidia. And to manage the disease, use the acid delinted seed and treat it with carboxin or chlorothalonil at the rate of 4 gram per kg, and remove and completely destroy the infected plant debris which harbors the pathogenic propagules, and apply increased dose of potash, which in turn with along with the balanced nutrition dose of nitrogenous and phosphatic fertilizers that will give the plant a tolerant capacity. And then apply more amount of organic manures, follow mixed trapping, and then grow some of the disease resistant varieties belongs to the Gossypium hirsutum as well as Gossypium barbadensis, like Varalakshmi, Vijay Pratap, Jayatar as well as Viram. And then besides those who are going for only the organic cultivation, they can go for seed treatment with the bacillus or trichoderma, as well as they apply the trichoderma basally at the rate of 2.5 kg mixed with 250 kg of well decomposed farmyard manure and apply in the field during sowing as well as 90 days after the sowing. And if the disease is very severe in the grown up plant, then go for spot drenching with the carbendazim, which is mixed at the rate of 1 gram per liter of water. And the next important disease is verticillium wilt caused by Verticillium dahliae, and the symptoms are seen when the crop is at the square or bowl formation stage. The first symptom can be seen as the bronzing of veins, which is followed by the intervenal chlorosis or yellowing. Finally, the leaves begin to dry and giving a scorched appearance. At this stage, the characteristic diagnostic symptom, that is drying of leaf margin and the area in between the vein, which shows a tiger claw like symptom, which is mentioned as tiger claw or tiger stripe symptom. And the affected leaves may fall and by leaving the branches barren. And when you take the longitudinal section of the affected plant stem, and it will show pinkish discoloration, what we have studied in fusarium, that is brown or black discoloration. But here in this case, due to verticillium infection, the internal portion as the vascular tissue become pinkish, and the infected leaf also show brown spot at the end of the petioles. And ultimately the affected plant may bear very small bolts as well as immature lint. This is the typical symptom that appears on the field, and this is the tiger claw symptom you could observe, that is intervenal necrosis of intervenal portions and then venal necrosis, which gives the tiger's claw like symptom. And this is the internal discoloration of affected plant, where it show pinkish discoloration of the vascular region. This is the healthy plant, while this is the infected plant, cross section of the infected plant. And the pathogen that produces two types of spores, the conidia, which are single celled, hyaline, spherical or oval, and bound singly on the verticillate conidiophore, while it produces microsclerotia, which are globose or oblong in shape and which remain in the infected plants. And the low temperature of 15 to 20 degrees Celsius and water stagnation and high soil moisture are favoring the development of disease. In the heavy soils with alkaline reaction favors the disease development. Heavy doses of application of nitrogenous fertilizers favors the development of verticillium wilt. This is the verticilloid arrangement of Verticillium conidia, and this is the presence of microsclerotia in the infected plant tissues. And the fungi infect some of the other host plants like brinjal, chili, as well as tobacco, bendi, etc. And it can survive in the infected plant debris as well as in the soil up to 14 years as sclerotia. The seeds also carry some of the inoculum. And the microsclerotia present in conidia, microsclerotia as well as the conidia, which get attached with the fuzz of the seed and through which it is carried to the field. And the primary spread is mainly through the microsclerotia or the conidia present in the soil, and then secondary spread is through the contacts of the root from the infected plant and dissemination of infected plant pots through the irrigation water as well as other farm implements serves as the secondary source of inoculum. And then you have to treat the seeds. Acid, we got acid deal in the seed to get away or to void off or to get away from the sclerotia as well as the conidia, which get attached with the fuzz. And this acid diluted seeds has to be treated with the carboxin or carbendazim at the rate of 4 gram per kg, and we have to remove the infected plant debris which harbors the inoculum, and then deep plowing in summer which helps in exposing the soil borne inoculum and then make it dry so that the infection ratio got reduced. And then apply heavy dose of organic manure which helpful in building up the beneficial microbial population. If the incidence is severe in a particular location, and then go for crop rotation by growing the non-host crop like paddy, lucerne, chrysanthemum, etc. in the grown-up plant if the disease incidence is more severe, and then go for spot drenching with the carbendazim. And growing of some of the resistant varieties is also recommended, which includes Sujata, Suvin as well as CBS 156, and the tolerant variety, that is MCU 5VT, which is also recommended. And for the organic management practices, you can treat the seeds with either Bacillus subtilis or Trichoderma aspergillum at the rate of 10 gram or 4 gram respectively per kg of seed, and then application of Trichoderma at the rate of 2.5 kg along with the 250 kg farmyard manure in the field before 15 days in the manure, and leave it for 15 days, and then apply to the soil at the time of sowing as well as repeat it at 90 days after sowing, which is helpful in reducing the disease incidence. And then the root rot, which is caused by Macrophomina fasciolina, the perfect stage is Rhizoctonia bataticola, and the fungus which causes 3 types of symptoms. First, in the case of seedling, it causes seedling disease, and then sores in symptom, and then root rot in the matured plants. And then the seedling, the germinating seedlings and the seedlings of 1 or 2 weeks old are attacked by this fungi and the hypocotyl region and causing black lesions, ultimately resulting in girdling of the stem and death of the seedlings, which cause large gappiness in the field. And this is the infection of Macrophomina fasciolina in the field, where we could see the large gappiness due to death of seedlings. And the source in symptom which is caused on the plant during 4 to 6 weeks after sowing. The pathogen known to produce dark reddish brown cankers at the stem near the soil level, and which turn into dark black, and due to infection at the basal level the plant breaks off at the point of infection and leaving the entire plant dry and completely dead after some time, and which is mentioned as shin symptom. This is the typical source and symptom produced on the infected seedlings. And the typical root rot symptom which occurs normally at the time of maturity of the plant, the prominent symptom that includes sudden and complete wilting of the plant in patches. Initially the leaves will droop suddenly and ultimately leads to death of the plants, and the affected plants, when you pulled, it reveal rotting of entire root system. It also shows shredding of barks at the basal portion, and on the shredded barks it produces black sclerotia. A large number of dark brown or black sclerotia are produced on the shredded barks. And this is the typical shredding of barks where you could see the formation of sclerotia. This is the typical shredding of barks due to infection, and these are all the black sclerotia produced by the pathogen Macrophomina fasciolina. And the pathogen which produces septate and highly branched hyphae and produces black irregular sclerotia. The dry weather followed by heavy rains and the soil temperature of 30 to 35 degree and the low soil moisture, which are all the favorable environment for the development of this pathogen. Besides, the cultivation of the favorable hosts like vegetables, oil seeds, and legumes preceding the cotton crop, and the wounds caused by the ash weevil grubs as well as the nematodes, all these are favorable for the development of this disease under field condition. This is the typical irregular black sclerotia, and this is the pycnidium produced by the Macrophomina fasciolina. The sclerotia is produced by Rhizoctonia pataticola, while the Macrophomina fasciolina stage, which produces the pycnidium, and hence the life cycle. It produce the pathogen could be identified both as on the pycnidial stage as well as on the sclerotial stage. The pycnidial stage is caused by the Rhizoctonia botanicola, Microphomina fasciolina, and the sclerotial stage, which is caused by the Rhizoctonia botanicola. This pycnidium is a flask-shaped structure which contains the conidia as well as the spores. And the soil-borne sclerotia, which lives for a longer period in the soil and helps in the spreading of infections. The irrigation water farm implements are also responsible for spread of this disease. And to manage this disease, treat the seeds with the trichoderma at the rate of 4 gram per kg, or Pseudomonas fluorescens at the rate of 10 gram per kg of seed, or the chemicals like carboxin or carbendazim. In the grown up plant, if the incidence is noticed, and then go for spot drenching with 0.1% carbendazim or 0.05% benomyl. Benomyl is not available in market nowadays. Apply heavy farmyard manure at the rate of 10 tons per hectare, and along with the neem cake. Adjust the time of sowing, that is the early sowing during the first week of April, or late sowing during the last week of June, which will helpful in the crop to escape from the high soil temperature period, and hence which can escape from the infection of fasciolina, Macrophomina fasciolina. And then intercropping with the sorghum as well as moth bean, which is lowering the soil temperature and in turn helpful in reducing the percent incidence under the field condition. And then for the organic practice, as we learnt in the previous disease, we can go for seed treatment with the biocontrol agents like trichoderma or bacillus, and then mass multiply the trichoderma asperellum mixed with the 250 kg of farmyard manure, and then apply at the time of sowing as well as after 90 days. But in the grown up plant, you can go for application of only carbendazim at the rate mixed at the rate of 1 gram per liter of water, or trifloxystrobin plus tebuconazole combination fungicide at the rate of 0.75 gram per liter of water, and drench at the base of infected plants. And then the next important disease is the anthracnose, caused by Coltotrichum capsicii, which produces small reddish brown circular spots on the cotyledons as well as on the primary leaves. The lesions may also develop at the collar region, stem, and ultimately leads to girdling and wilting, finally death of seedlings. In the matured plants, it attacks the stem and leading to splitting of stem as well as shredding of bark, and it also produces dark lesions or spotting on the bolls, which are small water soaked reddish brown depressed spots found on the bolls. Since it is producing the depression, the pathogen may also enter inside the bowl and stain the lints, and the affected lint may also turn into yellow or brown in color. This is the typical lesion formation on leaves as well as on the bolls. And the pathogen forms large number of acerulae on the infected parts. The acerulae, which in turn contains conidiophore, conidia as well as setae. The conidiophores, which are slightly curved, short and club shaped, while the conidia, which are hyaline, falcate shaped, single celled, and with this oil globules. Numerous black colored acerulae are formed on the center portion of infected center portion of the infection or lesions. And then prolonged rainfall at the time of boll formation and then closer planting are favorable for the development of disease. This is the typical establishment of acerulae on the infected plant parts, and this acerulae which contains, and the outer one is epidermis, and then this is the conidiophore which bears single-celled conidia. The pathogen survives as a dormant mycelium in the seed, and the conidia which are present on the surface of the seed which can be viable for at least an year, and the pathogen which also perpetuates on the in the rotten bolls as well as in the plant debris. The secondary spread is mainly through the airborne conidia. The pathogen also survives in some of the weed hosts like Aristolochia as well as hibiscus. And then to manage this disease, as we studied in the previous diseases, here also the acid delinted seeds has to be treated with either carbendazim or carboxin or thiram or capton at the rate of 2 gram per kg, and removal of infected plant debris which reduces the inoculum density in the soil, and rogue out the weed host which mentioned in the previous slide, so that the pathogen which harbored on the feed host would also get eradicated from the site of infection. And then spray if the disease is noticed at the later stage, and then spray either mangozeb at the rate of 2 kg or copper oxychloride at the rate of 2.5 kg, or Ziram 2.5 liters, or carbendazim 500 grams per hectare can be used for to manage the disease under field condition. And then the grey mildew which is also nicknamed as areolate mildew, which is caused by Ramularia areola. The disease usually appears on the undersurface of the bottommost leaves when the crop is nearing the maturity. It produces irregular to angular, pale, translucent regions which develop on the lower surface, usually bound by the veinlets. These lesions bound by the veins and veinlets, and hence it is mentioned as areolate mildew. On the upper surface, the lesions appears as light green or yellow green discoloration, frosty white grey powdery growth which consisting of conidia and conidiophores of the fungi appears on the lower surface of the leaf. When several spots coalesce together, the entire leaf is covered with white powdery growth and ultimately leads to drying of leaves. The infection spreads to the upper part of the plant also, and the entire plant will be affected. All the affected leaves are dry up from the margin, cup inward, and then turn yellowish brown, fall prematurely. And this is the typical grey mildew symptom found on the infected leaves. And this is the areolate powdery spots on the undersurface of the leaf. So in severe cases which may need to development of necrotic patches. And the pathogen which is endophytic and having producing septate mycelium. The conidiophores are short, hyaline and branched at the base. The conidia are bound singly or in chains at the tip of conidiophores, and the conidia are hyaline, irregular, oblong with pointed ends, sometimes with rounded or flattened end also, and mostly unicellular and sometimes 1 to 3 septate. And the perfect stage of the fungi that produces perithecia, which in turn contain assae and ascospores. The ascospores which are hyaline and usually two-celled, and this is the typical conidiophore of the fungi. And wet humid conditions during the winter are favorable in the development of disease, and intermittent rain during northeast monsoon and prevalence of low temperature 20 to 30 degree during October January, closer planting of the plants, excessive application of nitrogenous fertilizers, early sowing or late sowing of cotton crop, all are favoring the development of disease under field conditions. The fungi survives during the summer in the infected plant debris. The perennial cotton plants and the self-sown cotton plants harbors the pathogen during summer months. Primary infection is mainly through the conidia from infected plant debris, and secondary spread is through the wind, rain splash, irrigation water, and farm implements. To manage the disease, remove the infected plant debris which serves as source of inoculum, and then rogue out the self-sown cotton crop which harbors the infection. Avoid excessive application of nitrogenous fertilizers, as the excessive nitrogen application favors the disease development. And then spacing should also be adopted appropriately. In the closer spacing is also responsible for faster development of this disease. And then grow some of the resistant varieties like Sujata as well as Varalakshmi to get away from the infection. And for the managing the disease in the grown up plant, you can go for application of any one of the fungicides listed above, listed in the slide. And ultimately the tebuconazole group, which includes difenoconazole, tebuconazole or propiconazole, or the latest generation fungicides azoxystrobin, or the combination fungicide azoxystrobin plus difenoconazole, can be used at 60, 90 and 120 days after sowing, which will be highly helpful in reducing the incidence under field conditions. And then the bold rot, and which is caused by multiple pathogens, which includes fusarium, coltotrichum, aspergillus, flavus aspergillus, niger, rhizopus, and then nematospora and botrytis diplodia, etc. All these pathogens whatsoever present as a propagule in the air, which are get attached on the bowl surface and establishes causing the bold rot. And this is the typical bold rot which causes deal in the lint, internal lint are completely discolored and leading to premature opening of the bowl and internal rotting. And this is the spores produced by aspergillus, and this is also aspergillus, rhizopus, and this is the botrytis diplodia, and then fusarium, coltotrichum, etc. So initially it produces small brown or black dots on the bolls, and later it covers the entire bolls. The internal tissues are rotten, and hence the seeds are also be rotten, and the bolls will never burst open and they fall off, or sometimes it corresponds for immature opening of the of infected bolls, and the rotting affected from the pericarp and enter into the bowl and causes complete internal damages. Large number of fruiting bodies of the fungi are produced in the infected bowls, internal portion of the infected bowls. Heavy rainfall during the square as well as bowl formation stage, and then some of the damages or injuries caused by the insects, especially the red cotton bug, Disturgus singulatus, and then closer spacing and excessive nitrogenous fertilizer application, all are favoring the development of this disease. And the fungi which survive in the infected bowls, and the insects mainly helpful in spreading the infection from infected to the healthy. The fungi make their entry only through the wounds caused by the insects, and the secondary spread is through airborne conidia. And adaptation of appropriate recommended spacing is highly helpful to reduce the disease incidence, and with along with the application of recommended doses of fertilizers, and then spraying of the insecticides like fulminate, which is a combination pesticide, that is fenvalerate plus copper oxychloride or carbendazim or mancozeb, which will reduce the incidence. It has to be sprayed at 45th day and 15 days later. 2 to 3 sprays are required to reduce the incidence level. And then the leaf blight caused by Alternaria macrospora. This disease occur at all stages of the crop and more severe on the plants during 40 to 60 days old crop, in the 40 to 60 days old crop. Small pale brown irregular round spots are produced on the leaf, and each spot has a central lesions surrounded in a concentric circles. Several such spots coalesce together and form blighted area. The affected leaves become brittle and then fall off. Sometimes you may see the stem lesions in the infected plants. In severe cases, the spots may also appear on the bracts as well as on the boles. This is the typical spot. Initial spots produced as a dark brown and later enlarges and causes blights in the concentric circles, blighted area. Concentric circles in the blighted area. This is completely blight affected leaves, complete drying of leaves, and this is the infection by the alternaria on boles which are deep seated and enter inside the bole, causes damaging on the lint also. There are two different species that is responsible for causing this blight, that is Alternaria macrospora as well as Alternaria alternata, and the pathogen which produces dark brown short 1 to 3 8 1 to 8 septate irregularly bend or geniculate conidiophores with a single conidium at the apex. The conidia are obclavate, light or dark brown in color, 3 to 9 septation, and the unique character of alternaria, that is transverse septation. The spore is having both longitudinal as well as horizontal septation. High humidity, intermittent rainfall, and then moderate temperature of 25 to 28 degree responsible for the favorable for the development of disease. The fungi, the pathogen that survives in the infected debris as a dormant mycelium, and primarily it spreads through the irrigation water, and the secondary spread is mainly through the air. And to manage the disease we can go for application of the fungicides listed in this slide, which includes the difenoconazole, tebuconazole, propiconazole, and the azoxystrobin, the strobilurin groups like trifloxystrobin, combination of trifloxystrobin with tebuconazole, or application of some of the contact fungicides like copper oxychloride, chlorothalonil, etc. on 60th, 90th, and 120 days after sowing. And then application of Bacillus subtilis at the rate of 400 gram per acre on 60th, 90th, and 120th days after sowing is also recommended in case of organic cultivation. And the important bacterial disease in the case of cotton is bacterial blight, which is caused by Xanthomonas campestris pathovar malvacearum. It produces 5 different symptoms in the cotton plant, which includes seedling blight, angular leaf spot, veinal necrosis or vein banding, and then black arm of cotton, and then square rot or bold rot. And with the 5 kinds of symptom itself shows the pathogen which affects the plant from the seedling up to the maturity of the crop. During the seedling stage, the pathogen which produces a small water-soaked circular irregular lesions on the cotyledons. Later, the infection spreads to the stem also through the petiole and causes withering and death of the seedlings. This is the typical water-soaked lesions on the cotyledons, and this spreads, and the infection spreads through the veins to the petiole and then to the plant, leading to death of the seedlings. And then in some of the grown up plant it produces angular leaf spot, where the pathogen produces small dark green water soaked area on the lower surface of the leaf, which gradually enlarges, become angular in nature. That is restricted, the spot development or enlargement is restricted by the veins and veinlets, and the spots are visible on both the surface of the leaves. And since it is restricted by the veins and veinlets or within the angle in nature, it is mentioned as angular leaf spot. As the lesion become older, they turn reddish brown in color, and then it spreads to the veins and veinlets, leading to development of vein blight. And this is the typical angular leaf spot produced by the bacteria on the leaves. And in the third stage, that is the veinal blight or vein necrosis or black vein stage, the infection of the vein leading to blackening of veins and veinlets and gives typical blighting appearance. And on the lower surface of the leaf, you can see the bacterial oozes which form a crest or scale on the undersurface of the leaf. The affected leaves become crinkled, twisted inward, and show withering symptoms. And the infection also spreads from the vein to the petiole and leading to blighting and defoliation. And this is the venal necrosis or venal blight caused by the bacterial pathogen. And the infection which is angular in nature and spreads to the veins, which is clearly understood from this picture. And then the next stage is black arm. On the stem and the fruiting branches, dark brown or black lesions produced by the bacteria, which causes girdling of the stem as well as the branches and cause premature drooping of the leaves. Cracking may also be seen on the stem, and sometimes gummosis may be noticed, which resulting in breaking off the stem at the point of infection and hang typically as a dry black twig to give a characteristic black arm symptom. And this is the black arm symptom caused by this is the lesions formed on the stem, and this is the typical venal necrosis. This is the lesion on the stem, and this infection which spreads to the petiole and leads to black arm development of black arm symptom. And final one is the square rot or bowl rot. On the bowls, the pathogen known to produce water-soaked lesions turn into dark black and then shrunken and later become irregular. The infection on the mature bowls leads to premature bursting of bowls. The bacterium spreads inside the bowl and causes discoloration of lint or yellowing of lint due to development of bacterial oozes, and which ultimately results in reduced market value. And the pathogen also affects the seed and causes reduction in the size as well as the viability of the seeds. And this is the typical water soaked lesions on the bowls, and this is the bacterium, which is short, rod-shaped, single polar with a single polar flagella, gram-negative in nature, and non-spore forming pathogen. And optimum soil temperature of 28 degree or atmospheric temperature of 30 to 40 degree with relative humidity of 85%, and early sowing of cotton, and then delayed thinning, poor tillage, late irrigation, potassium deficiency, etc. All these are responsible for the development, initiation as well as development of the disease. These are all the favorable conditions. And the bacterium which survives in the infected plant debris as well as in the soil for some years. And the bacteria is also seed-borne in nature, which remains to form a slimy mass on the fuzz of seed coat, and hence the bacterium also attacks other hosts like Thunbergia and Eriodendron, Jatropha, etc. The primary infection mainly starts from the seed-borne bacterium, and the secondary infection is through rain splash or wind or implements or through the insects. So the mainly acid delinting, which removes the bacteria got attached with the lint of seed. And acid delinting we might have studied in some other courses also, that is mixing of concentrated sulfuric acid at the rate of 100 ml per kg of seed with constant stirring, so that all those first got attached with the seed are digested, turn into chocolate brown in color, and then subsequent washing with pure water to remove the acid fraction on the surface of the seeds, which is helpful in removing the bacteria present on the surface. After the acid delinting, treat the seeds with carboxin or oxycarboxin or even carbendazim at the rate of 2 gram per kg. And then soak the seeds in 100 ppm of streptomycin sulfate solution overnight will also be helpful in controlling the pathogen. And removal, as studied in many cases, the infected plant debris serves as source of inoculum, and hence remove and destroy the affected infected plant debris, and then rogue out the volunteer crop plants or cotton plants as well as the weed host, which serves as the harboring host. And then follow in severe case of infection, follow the crop rotation with the non-host crop. Early thinning as well as early earthing up will helpful in reducing the incidence level, and then growing of resistant varieties like Sujata 1412 or CRH71 are also advisable in case of severe infection. And in the grown up plant, if the infection is noticed, then spray streptomycin sulfate plus tetracycline mixture at the rate of 100 gram along with the copper oxychloride at the rate of 2 kg per hectare and or spraying of copper oxychloride alone at the rate of 2.5 kg per hectare, that would be helpful in reducing the incidence. And then the among the some of the minor infections caused by the Cercospora gossipina, which causes leaf spot disease, which produces round or irregular gray spots with blackish border and gray center. And the another important pathogen is Myrothecium, which is also causing some leaf spot symptom, Myrothecium roridum, which produces reddish spot which appear near the margin of the leaves, and the affected portions may fall off, leaving the irregular short hole symptom on the affected leaves. And then among the viral disease, important one is the tobacco streak virus. Initially, the virus which produces chlorotic yellow spot on the infected leaves, and the spot which later turn into necrotic a purple spot with yellow halo. The necrotic streaks will also be produced on the petiole as well as on the stem. The bud as well as the flower production are reduced in the infected plants, and the severe infection is also resulted in drying of squares. And this is the typical symptom necrotic spots. This is the chlorotic rings, complete necrosis of affected leaves due to the infection by tobacco streak virus. And the virus which survives in the alternate host, the primary spread is in the form from the infected plant, especially through the vector, that is thrips vector, thrips tabaci. And the pathogen which is coming under Bromoviridae under the genus Hilar virus, having the wide host range. For managing the disease, field sanitation, which will helpful in reducing the inoculum density, and then destroy the Parthenium in the cotton field, which serves as a harbor, which serves as a harbor for the insect vector. And then spraying of imidacloprid 17.8% SL at the rate of 125 ml per hectare, or fipronil 5% SC at the rate of 1500 ml per hectare, or flonicamide 50% WG at the rate of 150 gram per hectare are used to control the insect vector. So the second important viral disease is leaf curl disease caused by cotton leaf curl virus. Due to the infection, the leaves turn downward as well as upward and curled along the margin. The thickening of veins as well as formation of enation, especially on the main vein of the infected leaves, especially on the undersurface of the leaf. In severe case of infection, the leaves are completely curled and the growth is retarded. And the pathogen, which is cotton leaf curl virus, coming under Bergamo virus family, Bergamo virus category under the family Geminiviridae, and the virions are typical geminate particles, single strand circular DNA bipartite genome with the DNA A and DNA B components. And the primary source of this is the viriliferous insect vector, that is whitefly, Bimacea tabaci, and the alternate host and cultivated cultivated hosts may also serve as the virus reservoirs throughout the year. This is the typical cupping of leaves, upward cupping of leaf. This is the downward cupping of leaves. Vein thickening are seen in these two pictures. And this is the formation of enation, especially on the undersurface, especially in the main veins. What is enation? Enations are the small lobe or leaf-like structure formed on the undersurface of the leaf of infected plants. For managing this disease, planting date is to be adjusted so that the vector population get reduced, and then eliminate the volunteer perennial cotton crop or the alternate host, which including other malvaceous crops, helpful in reducing the insect population. And application of the biocontrol agent Bacillomyces farinosus, which parasitizes and reduces the vector population. And then spraying of neem extract at the, or 1% neem oil in reduces the infection up to 80%. And then vector management by using the chemical, that is the best chemical effective against the sucking pest. The example, the dimethoate, which can be applied at the rate of 0.2% to reduce the inoculum level. And then cotton stenosis or small leaf disease, which is caused by phytoplasma, and it appear the affected plant appears very stunted, and clustering of numerous extremely reduced leaf, and which gives the bushy appearance. The dormant buds are stimulated, which resulting in profuse vegetative growth, and the leaves are completely malformed and variously lobed. The flower remains small and with abortive ovary, and root systems are poor with a poorly developed root system. Rogue out the infected plant periodically, and then cotton varieties developed from Gossypium hirsutum and Gossypium barbadans are found to be somewhat resistant to this cotton stenosis or small leaf disease. And yet another one physiological disorder, that is magnesium deficiency, which completely leads to reddening of leaves, and for this you have to go for supplementing the magnesium in the form of magnesium sulfate, which will helpful in reducing the this type of symptoms, that is physiological disorder under field conditions. And that's all about the cotton crop. Thank you.